Hi class! For my project, I decided to learn about a geothermal spring habitat. I read a paper that was published about Hunter's Hot Springs in Oregon that described the different types of organisms occurring in several pools and springs and how they interact with each other. Hunter's Hot Springs is a well-studied alkaline geothermal spring in Oregon. The researchers in this study observed the different organisms in the pools and streams of the spring to determine the accuracy of previously determined environmental limits for the survival of each species in the spring. Geothermal pools and streams have gradients of both temperature and chemical composition that certain organisms need to survive, and the spring is divided into zones named for which species can survive within each section of the gradient. The ecology of Hunter's Hot Springs is relatively simple and includes several mat-forming cyanobacteria species that are visually distinguishable by color and morphological differences. The temperature range that these thermophiles in the spring can survive at is from about 30 degrees Celsius to about 74 degrees Celsius. Within this range, there are 4 zones with distinct communities including autotrophs, heterotrophs, and a species of microcrustacean that preys on certain microbial mat species. The researchers found that high temperatures were a limiting environmental factor for every species they studied, but that some species that are known to be able to survive in lower temperatures than they were observed to exist at in Hunter's Hot Springs were being limited by factors other than temperature at the lower end of their known temperature range. Some examples of these limiting factors include competition from organisms that could also survive in that range, but had other adaptations, such as motility, which allowed them to rise above and block sunlight from autotrophs that were not motile. Different growth habits, such as a crust which protects some species from intensive grazing, was also a factor. In this photo of the Grand Prismatic Spring at Yellowstone, you can see the differences in coloration, starting with blue in the center, where there aren't any large bacterial mats, and changing as the temperatures cool farther from the center. Each different colored area is a different thermal zone with a different community of organisms. Here is a chart I made of the different zones found in Hunter's Hot Springs. Water above about 74 degrees Celsius is too hot to support microbial mats that are visible to the naked eye. The hottest zone that contains these mats in Hunter's Hot Springs is from 55 degrees Celsius to 74 degrees Celsius, and it is called the Synechococcus chloroflexus zone. Synechococcus are a photosynthetic cyanobacteria that is the most heat-resistant organism in Hunter's Hot Springs. They form a green or yellow biofilm and thrive in a pretty wide temperature range. There are multiple species of Synechococcus in the mat, and the ones living in the hottest parts of their range were shown by DNA analysis to have evolved from the ones living at lower temperatures in their range. Below 68 degrees Celsius, another photosynthetic bacteria, Chloroflexus, grows with the Synechococcus. Chloroflexus is filamentous and makes orange mats beneath Synechococcus, where it can still do some photosynthesis, but also absorbs nutrients that the Synechococcus creates as a byproduct of its metabolism. At lower temperatures, where Synechococcus is no longer present, Chloroflexus grows without the biofilm on top, and these are the visible orange areas of the hot spring. Here is a picture of Synechococcus growing on top of Chloroflexus, and somebody has removed the top layer of Synechococcus so you can see the Chloroflexus underneath. At lower temperatures than the Synechococcus chloroflexus zone, we have our second zone, the Geitleranema chloroflexus zone. Geitleranema is a filamentous cyanobacterium that is motile, meaning it can move toward the light it needs to photosynthesize. It is dark in color and rises above the Chloroflexus and Synechococcus that is growing on the bottom of the spring. It also has the ability to contract when it's too bright to protect itself from too much sunlight, which exposes the bacterial mats below and allows them to get some light. Synechococcus doesn't do well having its light blocked by the Geitleranema, so it tends to be absent where Geitleranema is present. The presence of Geitleranema is a limiting factor that keeps Synechococcus from growing at lower temperatures in this spring, even though it can technically survive in those temperatures. In this photo, you can see some Geitleranema where it is growing above the orange Chloroflexus. This Geitleranema is in its contracted state, revealing the chloroflexus that would otherwise be covered up. Beside the chloroflexus, you can see some synechococcus as well. This synechococcus is closer to the center of the pool where it is too hot for the Geitler-enema and the chloroflexus. The next zone is called the ostracod Pleurocapsa calothrix zone, and it's my favorite one because it has predators. Below 48 degrees Celsius in Hunter's Hot Springs, there are microcrustaceans called ostracods. These little guys eat the Geitler-enema and are so efficient that practically none of it grows within the temperature range that the ostracods find ideal. Ostracods are also known to make excursions into hotter areas to graze the Geitlerina there, but they can only last about 20 minutes before they are rendered comatose by the heat, and the current carries them back to cooler temperatures where they can recover. Because the Geitler-enema can't grow where so many ostracods live, an environmental niche is opened up and it is exploited by a mat composed of two photosynthetic organisms, Pleurocapsa and calothrix. Pleurocapsa and calothrix grow together in a tough leathery mat that ostracods are unable to graze on. The Pleurocapsa calothrix mats are dependent on the ostracods to clear the space they need to survive. To the left is a photo of an ostracod under a microscope. They are actually so small that they look like tiny white dots, barely visible to the naked eye. On the right is a photo of the symbiotic mat made up of Pleurocapsa and calothrix. The last zone is called the sulfate-reducing zone, with temperatures between 30 and 40 degrees Celsius. There is a purple flagellated bacteria called thermocomadium that have elemental sulfur inside their cells and do photosynthesis without oxygen during the day, but at night they rise to the top of the water column and use oxygen and sulfur for chemolithotrophy, a process which allows them to get energy from inorganic compounds instead of sunlight. Another resident of the sulfate-reducing zone is Beggiatoa, a chemoheterotroph that oxidizes hydrogen sulfide for its energy source and derives nutrition from eating organic matter. This zone also contains Geitler-enema, since the ostracods that graze on them at higher temperatures cannot survive in an environment with so much sulfur. For the community outreach portion of my project, I took a group of people to Grover Hot Springs State Park. I went over the information that I've just presented to you, and we set out to look for some of these zones. We used a thermometer to measure the water temperature in different areas of the hot spring and took pictures of what we found. We were able to find plenty of what looked like synechococcus and chloroflexus, but there were also some other things growing there that didn't look like anything from Hunter's Hot Springs. We also brought a hand lens in case there were ostracods, but unfortunately we couldn't find any. I asked my group what they liked the most about the presentation and what they would like to learn more about. Based on their feedback, if I were to give this presentation again, I would talk more about the history of Grover Hot Springs, the nutrient and sulfur cycling, and try to take the group somewhere where there are ostracods. It would also be really cool to have a microscopy section of the presentation so we could see what the mats look like up close.